Good afternoon, friends. Welcome to our Ask Me Anything session that's being hosted by AEA. We're opening the transmission or the recording a little bit early, the webinar a little bit early to allow everyone to come in. And if you would like, we have a couple of questions that have come in advance where people emailed in questions that we can start from, but I would also suggest if you have any questions, anything, I really rely on the audience to help guide the conversation, lead the conversation. So if there's any questions you would love to have answered, type them into the Q&A box. And I should just clarify, specifically the Q&A box and not the chat. Q&A should be the furthest right on your device. I think that's the case for most devices at least. And that helps me to keep track, because with the chat, there can be lots of back and forth between audience members, and I can lose sight of the questions that get asked really quickly. So thank you for that. And I see quite a few people have joined already, and we're at the hour, so we're going to get started. So let me see here. There's a couple of questions that have come through on email that we'll start off with as more questions come in. I see there's already being populated pretty quickly. The first question is from Ian Thomas. And the question is, some big names still commonly claim that humic acids are a fungal food and should be included in biological brews as a food and stimulant for fungal populations. You have spoken very accurately about humus being the end product of decomposition, and therefore the concept of humic acid as a food is incorrect. Could you please elaborate on this and explain the actual benefits of using humic acid? Is it that these acids are more of a conditioner that help conditions for fungi to thrive rather than a food source? And then there's other questions as well, but that's the first question. So where is it? Here we go. This is the book that everyone needs to, that every farmer, everyone who's a serious student of organic substances and building soil organic matter needs. It's about a $150 book. It is sometimes difficult to buy on Amazon, but if you go to William Jackson's website, I was very disappointed. I really wanted to interview Dr. Bill Jackson on the podcast and record some of his information, some of his knowledge. And it was, unfortunately, he passed away before I was able to do that, just fairly recently in the last year or two. But this book is, the full title is Organic Soil Conditioning, and the subtitle is Humic, Fulvic, and Microbial Balance. It's an expensive book. It's $150 or something like that, used to be anyway. And somehow as a culture, we have developed this idea that if books are really expensive or carry a big price tag, that we're going to avoid them. And information and knowledge is one of the most valuable things that you can have. Many of you know that I've very highly and frequently recommended Don Huber's book, or not just Don's, but Lawrence Stanton, the book Mineral Nutrition and Plant Disease. It's also a book with a significant price tag, roughly $100. Scholarly book. This is also a scholarly book, lots of references. It is older. Looking to see when it was published. It was published in 1993. So there's a lot of new research that has come out since this book was published, but this is still the definitive reference that I'm aware of, at least, on humic and fulvic substances. And so I highly recommend it. I think more people should get it and become familiar with some of the things that he talked about, because it is in this book that I first connected the dots and to understanding that the development of humic substances in the soil profile is a function of the fat content, the lipid content in plants. And this was published in 93. It's been out, the information has been out there for a long time, but people aren't reading it, and which is a shame, because it is incredibly valuable. So to answer the question directly, as you can imagine by the size of the book, about 4 inches thick or 3 inches thick, maybe, the benefits and the actions are multitude. It's much more than I can elaborate in a brief response. But my understanding is that they are a combination of a structure that these humic substances provide a structural home, if you will, that really stimulates biology. And they also, in the case of fulvic acids, they improve mineral absorption and mineral transport and mineral absorption across cell membranes. So there are lots of nutrient interactions and microbial interactions that, well, let's just say maybe the best way to describe these materials as I'm thinking about it, the word that comes to mind is a catalyst. We understand that enzymes are catalysts and speed up chemical reactions. And perhaps the best way to describe fulvic and humic materials is that they are a catalyst and they speed up the development of microbial processes and nutrient interactions with microbes. And that is where a big part of where their value comes from. The second question from Ian is, you mentioned that all trace minerals are better applied in the reduced form. Do you see any merit in applying them in the oxidized form? For example, applying manganese sulfate if a reduced form is not available. So I should just clarify that the correct way to state this would be to say that I believe it is most beneficial for plants to absorb nutrients or to apply nutrients in the form that plants absorb them in. So there are some nutrients like zinc, for example, that only has one oxidation state of the +2 form. And so in the case of zinc, applying zinc sulfate is can be a great application, particularly the soil. And I actually think I've made this comment before, but I think that this is one of the reasons why zinc applications are relatively popular compared to manganese or iron, because mainstream growers who have really compromised soil microbial communities and compromised soil health can apply zinc and observe a crop response. Where if they apply manganese or if they apply iron, particularly to the soil, they frequently fail to observe a crop response. And it's not because the crop doesn't need manganese or iron, it's because the crop doesn't get the product that was applied because of soil imbalances. So in answer to the question, I'll just say that our observation, our experience has been that when we apply minerals to the soil, just for the record also, manganese sulfate, the manganese in manganese sulfate, in my understanding, is actually in the reduced form. So I'm not a chemist, but that's what I've been informed, is that the manganese in manganese sulfate is in the reduced form. But then when you apply it to soil, and that soil is oxidized, it rapidly converts to the oxidized form. When you apply manganese sulfate to the leaf surface, and it's exposed to the atmosphere and to air, to oxygen, then it also rapidly oxidizes and the plant fails to absorb it efficiently and effectively. So I missed completing my prior thought, which is that in our observation, when we apply manganese sulfate to the soil, we see the soil analysis on a typical ammonium acetate or a DTCPA analysis. We see the soil analysis increase. So the numbers on the soil report go up. The numbers on the SAP analysis have not ever increased that we have observed from putting on soil applications of manganese and iron sulfate. Actually, I shouldn't use the word never, because there have been a few exceptional occasions where when the soil was really reduced, so if you had soil that was saturated for several weeks after application because of high rainfall or whatever the case might be, in those environments, in those cases, we did see increases of manganese and iron absorption from when applied to the soil. But those situations are relatively rare. Next question is there anything that can be done at home to easily reduce the oxidized form of these mineral amendments where these are all I have available to me? could anaerobically fermenting them achieve this? And the answer is yes. So we have, I don't have a lot of experience or observation with this, but I've heard anecdotally from growers who applied manganese sulfate or iron sulfate or whatever into bokashi and had an anaerobic fermentation or composting process, if you want to call it that. And upon application, they observed very nice crop responses of increased manganese absorption and iron absorption and so forth. All right, so I'm going to go to some of the questions that have come through. Question from Marcus: when applying a soil amendment in a liquid form on a pasture, how much will plants take up through their leaves? My goodness, that's a question. It's a very big question because soil amendment can be many different things. And obviously particle sizing and all of that plays a role. But in general, I would say that if the product that we are applying, for the sake of conversation, a soil amendment would commonly be a manure or a compost or a rock powder. So I'm going to assume that we're talking about rock powders, whether that be limestone or gypsum or rock phosphate or whatever the case might be. If we're applying liquid lime and we have relatively small particle sizing, 200 to 400 mesh or less than 50 microns, plants can absorb some of those particles through the leaf. And there are various mechanisms. It is not just through the stomata. Plants do absorb larger particles through endocytosis and through other mechanisms other than just through the stomata. But the absorption through the leaf is going to be relatively small, and it's going to vary based on the product that we're talking about. But I would anticipate less than 2 to 3% of what lands on the leaf surface is actually going to be absorbed by the leaf for those types of materials. Question from Timothy. Could you comment on municipal compost facilities using biosolids, the use of this compost in agriculture, and the potential for PFAs and other chemical contamination in farming fields? I've considered having a guest on the podcast about this topic. Let me see. It's nice to have my bookshelf right behind me. David Lewis, in the book titled Science for Sale, is a book that anyone who has interest in learning about biosolids needs to read, and definitely anyone who is interested or is considering using biosolids needs to read. The short version that you can probably guess even from without reading the book is that biosolids have been very well documented through peer-reviewed research to be very hazardous for your health, and to be very hazardous for soil's health, and to be very hazardous to the health of all the neighbors in the surrounding areas where it is applied to the fields. So my recommendation would be to avoid at all costs. Question from Peter. We are being prepared by the industry. I like the beginning to that sentence. We are being prepared by the industry. That's very, very true. Far more often than we want to acknowledge or realize it's in a sense, I made the comment on a recent podcast interview with Jason Malk that of the people who are farming the farmers, and that phrase has become popular beyond what I ever anticipated. But it's very common that there is this cultivation of fear that occurs in advance of a potential problem to sell solutions that fit a certain narrative. We are being prepared by the industry that tar spot on corn is going to be a major problem here in Ontario next year. I have not applied any fungicide post-emerge on corn and soys for many years and did not want to start again. What can I do to mitigate this disease? Trying to recall, we actually had a conversation internally with the AEA consulting team, I remember there were some message exchanges about tar spot where one of our team members reported having success just with using the standard AEA treatment protocol. And if I recall correctly, the comment was made that just applications of BioCoat Gold as an inoculant of a probiotic was the growers who applied that did not have tar spot in those fields. So of course, BioCoat Gold is not a fungicide, and we can't say that it can be used to treat a disease. But based on the experiences of some of our agronomists, it's possible that it can be used to prevent the emergence of tar spot. So that was, and I would suggest reaching out to the AEA team for more information on that, because I was just an observer in an online conversation, did not actually participate. But I would say that for most diseases, it is a wiser course of action to avoid prophylactic treatments. Wait until there is actually a problem before you put on a fungicide application. And in the majority of cases, you will find that the fungicide application is not actually needed. And there's a very important reason for this, because in many cases, a fungicide application, well, not in many cases, in all known cases, fungicide applications will change a plant's biochemistry and increase its susceptibility to the disease that you are trying to serve as a preventative against. So it actually, a fungicide application as a preventative is an oxymoron, because you do not prevent fungal infections with fungicides, because they actually, as soon as the fungicide wears off, you have dramatically increased that plant's susceptibility to whatever you were trying to avoid in the first place. The question from Jake is, if no-till is ideal for soil health, what about deep ripping for vineyard preparation? And Jake, your question begins with an assumption. If no-till is ideal for soil health, and I would suggest that no-till is not universally ideal for soil health for all soils all the time. So if soils have been very compacted or have been historically mismanaged, then deep ripping can be a useful tool to mitigate that historical damage, as long as we then make sure to use management practices to not recreate the same problem that was there originally. So Don Huber makes this point as well when he talks about developing disease suppressive soils. No-till, I think, is, well, just in general, we should not permit idealism to take the place of practicality. We have to produce practical, real-world results. And in many soils, in many contexts, in many environments, no-till is a worthy goal that we should strive for and should achieve. But in some ecosystems, it doesn't make sense right from the get-go. Question from Marcus. What effect does soil aeration, such as with a yeoman plow, have on nutrient availability? Context is that there's no prior sap analysis on this property. The new leaf that grew after yeoman's levels of calcium are down and the boron is up. Well, again, this is a very open-ended question with lots of possible context, because it's not just about soil aeration, but it's also about what is the water availability, moisture availability, and so forth. So in general, when you aerate soil and you increase gas exchange, that increases oxygen flow into the soil, and that tends to have an oxidizing effect. In some soils that are compacted, that can be beneficial. And in some soils, that can have a negative effect. So I would say that in most soils, oxidation tends to change the soil microbial community. And this is, again, speaking in broad generalizations, but for most soils, that tends to produce a disease-enhancing soil microbial community rather than a disease-suppressive one, unless that soil was really compacted, and then it can actually have the opposite effect, a beneficial effect that you're looking for. Question from Anthony. Hi, Anthony. Are there any nutritional strategies for tilting plant energy to growth instead of reproduction for new orchard trees, yes, the foundational strategy is to maintain abundant levels of available calcium in the soil. So this is not about foliar applications of calcium, although those are beneficial, so much as maintaining very high levels of available calcium in the soil. And then also partnering that with high levels of cobalt in the soil as well. And this cobalt can also be foliar applied. And I'm, of course, basing this on a foundation of managing everything else. So it's not just those two, but having a particular emphasis on those two once you've addressed all the other bases of managing magnesium, phosphorus, potassium, etc. When we have covered all of our bases and everything else is taken care of, and we then amplify the available calcium in the soil profile, that will have the same effect on plant growth as applying large amounts of nitrogen fertilizer, except that instead of having widely spaced nodes, we'll have very tightly spaced nodes that on orchard trees might be 2 inches apart instead of 6 inches apart. So it's possible to produce a lot of vegetative growth that is very high quality, high carbon wood with narrowly spaced nodes with that abundance of calcium. And an example, again, this would have to be adapted for every environment, but an example of how we've managed that is one orchard comes to mind that we were asked to produce this effect on. We applied a total of 6,000 pounds of gypsum per acre over the 18-month period before planting the orchard, and produced very high calcium concentrations within the mound of the tree row. Now, that was specific for that soil environment, and those amounts may not need to be nearly that high for different soils and different contexts, but that's was the tool, the product that we used, and the application method that we used to bring calcium availability up to that level for that one farm that we were working on. Question from John on Spectrum Soil Inoculant. I have a little bit of a problem with the math on this product. An acre has rounded 6 million square inches. And so I think the math works out to roughly 8 bacteria per square inch. Is that sufficient to change the soil microbiology? What is the doubling time of these bacteria? And in a perfect world, what should be the concentration of the most abundant microbe per gram of soil? John, this is a great question. And it's a great question because it identifies one of the ways that we have been led to think about microbial inoculants incorrectly, from an incorrect premise. So we know that bacteria and organisms in the soil exchange genetic information extremely rapidly. Stephen Herod Buhner, in his book, which of his books is it? I think it's the one titled, it might be The Lost Language of Plants, if I'm not mistaken. He describes how when bacteria are exposed to an antibiotic and they develop antibiotic resistance quite rapidly in the soil profile in a matter of, I'm just going from memory here, so don't hold me to this, but I want to say it's something like 10 to 14 days. It's relatively quickly. Then, and this research was specifically on single strains or single species of bacteria. They will develop antibiotic resistance within a matter of a few days to a few weeks. But then that genetic information about how to be resistant to this group of antibiotics is shared with the rest of the microbial population of the soil profile through this process known as horizontal gene transfer. And so there is this rapid exchange of genetic information where all of a sudden the great majority, perhaps the entirety of the soil microbial population now has this genetic information and now has the capacity to be resistant to this group of antibiotics. And this is rather, if you think about it, really has the potential to be rather scary from the perspective that we have antibiotic exposure to the majority of our soil microbial communities, any that has had manure applied where animals were fed antibiotics does now have this antibiotic resistance. It's quite well documented. So we should not think when we apply soil inoculants, whether that's in the form of compost tea or a purchased inoculant or mycorrhizal fungi, whatever the context is. Well, actually, mycorrhizal fungi are a bit different because there we are looking for specific species. There are cases where we are looking for specific species and specific colonization. There are also many cases where, particularly in the context of bacterial applications, we are not looking so much that we apply a species and it establishes significant colonization and a large population displacing other microbes throughout the soil profile. In some cases, that might be what we are looking for. But from my understanding, in many cases, what we're looking for is we are looking that we have a strain of organisms that has a certain genetic capability. Maybe they have the capability of extracting and solubilizing phosphorus and making phosphorus available, or fixing nitrogen from the atmosphere, or whatever the case might be, specific disease suppressive pathways. What we are looking for is for those organisms that are added to the soil profile to share that genetic information with the rest of the soil microbial community. And that's really the way that we should think about microbial inoculants, is not about the addition of species so much as the addition of specific genetic information, because that's really how these microorganisms work and function and thrive as communities. There's this constant free flow and exchange of genetic information. And that's really how we should think about these microbial additions that we're making to the soil profile, particularly in the case of bacteria. So a question here from Elmer. Clover grass mixtures are nice forage and give the soil some rest between small grains and vegetables, potatoes. How can we terminate them or any other wintergreen cover crop in a biological, healthy, and organically legal way to make room for the next crop? Oh, yes, the conundrum that we all are trying to figure out. Here in the US, and I presume in Sweden too, although I don't know the answer, it is not organically legal to terminate with boric acid, and other forms of boron are not particularly effective in my personal observation and experience. There are getting to be more products available, like high doses of acetic acid, glacial acetic acid, and so forth. So there's different. I'm trying to think of what else to glacial acetic acid. Also, you could combine actually one practice or product or tool, whatever you want to call it, that has been effective in my experience in most situations is a combination of acetic acid and sodium chloride. Is that organically legal? In most cases, yes. I wouldn't know for sure about yours, but and it's also worth mentioning that that is only a burn off. It will burn off the top, but not completely kill the crop. So I'm sorry I don't have a better answer for you. That's the best answer I have at the moment. Question from Eugene. Is trichoderma a good idea to change the biology in a plum orchard with wet clay? Maybe. I don't know the answer to that. There is this narrative that trichoderma can be so aggressive that it excludes other biology. And I have some very significant question marks around that narrative, because my limited observation of trichoderma has been that it behaves as a pioneering species. And yes, it may be dominant in the short term after application, but in a period of perhaps months or years, it is no longer as dominant a species, and it makes way for other biology as well. I would suggest, though, that it is best. I've talked about our idea of synergistic stacks, that we don't apply just a fungal inoculant, but we combine it with other things. So think about applying a combination of trichoderma plus mycorrhizal fungi plus bacteria plus humic substances, and put together this combination that has the potential to behave in a synergistic way. And there is a very high probability that you will get significantly better results than just with the trichoderma itself. Question from Marcel. Marcel, hi John, how do we control powdery mildew in strawberries and apples? I don't know the answer to this question off the top of my head. What I can say is that in our experience and actual practice, we use sap analysis to balance nutrition, and powdery mildew tends to go away. Not just tends to go away, it does go away. Why? Is it because we have higher levels of manganese and zinc and iron and copper and boron? Maybe. Is it because we have higher levels of calcium? Maybe. I don't know. I don't know specific interactions that are in play there. All that I can say with a broad generality is that as we manage nutrition and optimize nutrition for plant health, powdery mildew certainly disappears. Question from, I'm not going to attempt to butcher this name. My apologies. Why is fruit fly becoming more of a menace? And we are unable to control their damage. What is the way forward? Fruit fly, any pest, the context here is that fruit, the reproductive parts of a plant, are often nutritionally the weakest parts of the plant, simply because they require the highest nutritional concentrations. The nutrient concentrations of zinc and manganese and copper and boron and other elements are often an order of magnitude or more higher in fruits and berries and seeds than they are in the leaves, which means that if there is any imbalance present, if they are low in one or another nutrient like zinc or manganese or boron, that deficiency shows up much more severely in the reproductive parts of the plant than it does in the vegetation. And because of that, because of this difference, fruit and seeds are often the last location on a plant to become fully disease resistant and insect resistant. So the pathway forward is exactly the same as it is for managing, for preventing other challenges and disease and pest challenges. And that is to balance nutrition, to optimize plant immunity. And I know that's a very broad general answer, but that's the best answer I can give you. And when that happens, you will have resistance to fruit fly. And just a personal anecdote, a recent experience with this, my father and my brother are both raising black raspberries, which are very susceptible to the SWD, spotted wing Drosophila. And their black raspberry patches are separated by approximately 500 yards, 400 or 500 yards distance. And one of them is consumed this last year's production season. One of them lost most of their crop to spotted wing Drosophila because the blackberries or black raspberries were secondary for them to their primary job, and they didn't take care of them. They didn't manage the nutrition well. They had major problems with SWD. 400 yards away, the other black raspberry patch had no SWD. It was non-detectable. They tested berries multiple times and couldn't find any. And the only difference between the two was some geographical distance and nutrition management. So it is possible to produce resistance to the fruit fly by managing nutrition. Question. Have you ever observed if foliar sprays of phosphate, such as monopotassium phosphate, increase resistance against powdery mildew in practice? Literature suggests that this might trigger phosphate-induced resistance, but I don't know of anybody doing this in practice. Have I ever observed this? On a very few, very limited occasions. And I think The reason it is not more consistent is because of other nutrient interactions that are present. So phosphorus is important. Phosphorus is very important for powdery mildew reduction, but so is manganese. And if I'm not mistaken, so is copper. So in some cases, if plants have adequate levels of manganese or copper or other of the elements that are required, but they're low in phosphorus, a foliar application of MKP might be very effective. But in cases where you have adequate levels of phosphorus, but low levels of manganese and copper, an application of MKP might be substantially less effective or completely ineffective at all. Question from Jake. I am a new vineyard owner trying to get up to speed with AEA and regenerative ag. One question relates to spraying inputs versus drip fertigation. What are the differences and the benefits of one over the other? So the differences are, I'll speak here in very broad generalities, and it may vary, your mileage may vary with your specific operation, but in general, if you are properly set up, fertigation takes a lot less labor than foliar spraying. And for many farms, that's a significant and a growing consideration. Dependent on soil and soil management, it can often take as much as 2x a product volume per acre to, and I'm not talking about in the row acre, I'm talking surface acre. It can take as much as 2 times higher product application in the fertigation to produce the same result as you produce with a foliar spray. And in regards to timing, if you put on a foliar spray, we expect to see nutrients increase in the sap analysis in a period of 2 to 3 days or less. And with a fertigation application, it can take a week to 10 days because you have the whole microbial population to take into consideration as well, and the dynamics that exist there. Those are the really big picture variables that come to mind right off the top of my head. Increasingly, operations who have challenges with labor availability are opting and are finding it less expensive to simply put on higher applications in the fertigation system than to put on foliar applications. There are trade-offs, and sometimes there is still the need for foliars in some situations. But in general, I would say both of them can be effective and can work. Question from Davis. I've heard you say in other webinars that you use a geological soil testing service to see what elements are in the soil in the total pool. Where can I get such a test? There are 2 laboratories that specialize in these tests. Both are Canadian. One is called Acme Labs, ACME. A second is called Agent Labs, AGAT. These labs are both, speaking plainly, they're both a bit of a pain to deal with. They focus on doing geological assays for the mining industry, and they have all types of test variations, and it can be difficult to decipher exactly what it is that you're looking for. And I don't recall what the test designations are off the top of my head. Many growers in the US at this moment are using Midwest Labs, and Midwest Labs has a specific test. They do this geological assay, and it is not advertised. It's not widely known. There's a specific test designation. I'm trying to remember what it is. Is it a P2 or a PS2? We may have to get that information and share it with you or attach it to the notes here on the webinar, because there's a specific designation that is not widely known, but it is available, and Midwest Labs does offer that. Question from Marcus. My goodness, we're 41 minutes in, and I see that we have 42 questions remaining. So I do like to offer some explanation, but I will try to be brief with the remainder of my answers to see that I can answer as many as possible. Question from Marcus. In high temperature conditions, anything above 76 degrees Fahrenheit, minerals start behaving differently as the plant goes into photorespiration. I assume that this only happens for the hours of the day that the plant is in the respiration phase. What are the plant's capabilities to correct this once there is not enough sunlight left for photosynthesis? Um, the capabilities to correct any imbalances that occurred during that period are dependent on the plant's carbohydrate reserves or energy reserves. Actually, the right way to say it would be that they're dependent on the plant's energy reserves. So those energy reserves are not necessarily just in the form of carbohydrates. They're also in the form of lipids and to a lesser degree, dependent on the levels of proteins as well. Obviously, we don't want plant to degrade proteins, so we don't want them to use proteins as an energy source. But the energy source that we do want them to use is lipids and to some degree sugars. So if you have a high energy plant, let's say a plant on level 3 of the plant health pyramid that has high lipid concentrations, then first of all, they will experience less stress during the photorespiration period. And they also have these energy reserves that they can pull from to reverse any potentially negative interactions that occurred during the photorespiration period and recover from those. So the answer to your question is the higher energy a plant is, the more energy reserve it has in the form of lipids or carbohydrates, the more rapidly it can reverse any oxidative stress or anything that happened from that photorespiration period. And the second part of your question is, how does this change the way we look at sap analysis, considering we're taking leaf samples in the morning when the leaf temperature is below the threshold, but during much of the day, the temperature is above this? Marcus, I really like the questions that you're asking. These are great questions that I've actually not thought much about. The kind of the immediate and obvious answer that comes to mind for me is we may want to look at nitrogen metabolism differently. I would guess the first place that we would see changes and variation would be in total nitrogen versus nitrate versus ammonium ratios. We may have higher levels of ammonium later in the day, possibly also higher levels of nitrate in the morning. So those ratios may shift and change a little bit. And there's also a possibility that sugar levels might change as well and fluctuate through the day. So that would be my first guess of where I would look, but we haven't actually tried measuring later during the day in these high stress periods. So I don't know the answer for sure. Question from Angelo. I have a question regarding apples. Since I've been using your products, the apples have been phenomenal, but what I'm currently encountering is that just prior to full maturing, the stem of the apple dries out and falls off. Have you encountered this before, and could you elaborate on this? Yes, we have encountered this before. And Angelo, I'm not familiar with your specific context of where you are in the world and what your management practices are. So I hope that my observations are appropriate for you. But when you have early fruit drop, premature fruit drop, this is a result of the plant accumulating abscisic acid, otherwise known as ABA. And abscisic acid first is produced naturally as a part of the maturing and senescence process. But as you are observing, your specific problem is that it is happening too quickly, too fast. And we see this happen. It's a self-inflicted problem by many growers. And they inflict this problem in two ways. One is they actually apply abscisic acid or ABA as a coloring agent, as a ripening agent to increase the coloration of the fruit. So yes, it does improve the coloring and it increases fruit drop at the same time. It's much wiser to improve fruit coloring by managing nutrition, and you don't get the excessive levels of ABA and fruit drop. The second way that growers inflict this problem upon themselves is by inducing drought stress or water deprivation. When you remove water from a tree crop, or from any plant for that matter, one of the well-known mechanisms is they increase ABA production, they increase abscisic acid. So doing any one of those two things, or particularly doing both in combination, is a recipe for premature fruit drop. And in my opinion, very unwise agronomic management. So that would be the first two places that I would look. And other levels or other types of stress as well, the different types of stress will increase the production of abscisic acid. And it's really abscisic acid that is, I mean, it has the name abscisic acid because it causes abscission, because it causes the separation of the stem. Question from Sergio. Is there a correlation between disease resistance in the plant leaf and in the fruit? I asked this because this year I noticed that I had perfectly healthy leaves without fungal pathogens, and in the same vines, I had bunches infected with powdery and downy mildew. This is an awesome observation, Sergio, and it matches our observations as well that I alluded to earlier in this webinar, where I talked about how often frequently the fruit, the berries, are the last location on a plant to become fully resistant, simply because they require higher nutrient concentrations than elsewhere on the plant. And because of that, they are often the first location for a nutrient deficiency to show up. Or I should say, nutrient deficiencies show up in a more pronounced manner in the fruits and the berries than they do in the vegetation. So this is a common observation. Is the same true with insect susceptibility? The answer is yes. In vineyards, is the ideal pH 6.4? Because in my SAP analysis, it is always between 3.5 and 4.4. Also with the other growers, I know that also that makes SAP analysis regularly. The answer to your last question, is the ideal pH 6.4? We learned from Bruce Tainio, who was a savant level genius. He indicated that 6.4 was the ideal pH for all plant species, a plant sap pH. And he was arriving at that number from a mathematical calculation based on plant frequency, vibratory frequency, and insect and disease susceptibility. From the work of Olivier Husson, we are learning, there's several interesting papers that are in the peer review process that will be published at some point in the near future, hopefully. The short answer is that phloem pH and xylem pH are usually dramatically different. One is quite alkaline, the phloem, if I'm not mistaken, and the xylem is often quite acidic. And so when we begin separating those out from a research perspective, first of all, it's really exciting because it allows us to, now that we've separated out phloem sap pH and redox environment versus xylem sap pH and redox environment, we're able to correlate to disease and insect susceptibility very accurately. And so my understanding, the way that I would communicate it at this moment, is that the collective tissue pH of a plant leaf is optimal at 6.4. But we don't know for certain what fraction of those total fluids that are in all these different leaf compartments inside the cell, extracellular fluids, xylem, phloem, and so forth. It's not clear how much of the sap that is extracted for a sap analysis comes from each of those segments. So while I am persuaded that the 6.4 pH number of the total leaf structure is quite likely to be accurate, there's a possibility we may need to revisit the desired value of sap pH in the sap, particularly because we don't fully understand where that is being drawn from inside the leaf. And so what we need to look at from a very practical perspective is to say, okay, for all of these sap analysis samples that we've collected, where are the growers and where are the blocks in the fields that have demonstrated true disease and insect resistance, and where are those sap pH levels? So that's something that we need to revisit in the future. Why can I not give short answers? I think details, and in many places, in many aspects of agriculture, details are important, but I now have more answers than I did 10 minutes, or more questions than I did 10 minutes ago. Question from Nabil: how can you see the liability of promoting regenerative practices which might cause some loss to some farmers, especially pest control by attrition? The variables are so huge, as I'm sure you are aware of. Well, let's start from the beginning. We do not observe any liabilities because we never tell growers to stop the use of all insecticides and pesticides and fungicides. Never. We work with very large-scale growers who have a lot at stake and a lot at risk, and we cannot. It would incur a huge risk on the grower's part for us to make them for us to make that recommendation for them. It would be, quite frankly, stupid. And so our pathway is, in effect, to say we believe this is possible. We have to earn the right to discontinue the use of pesticide applications. And then some growers have varying degrees of enthusiasm and appetite for risk. There have been many cases where we've told growers, we think this is possible. This is the road we want to go down. We want to manage plant nutrition in such a way to produce disease and insect resistance. And we think that a couple of years down the road, you will no longer need to use these pesticide applications. And there have been a number of occasions where at the end of a growing season, growers will tell us, oh yeah, by the way, we never applied any insecticide on that block and that we were doing your trial on, and we didn't have any insects. And we don't even find out about it until afterwards. And then there will be other situations where we go down the road 2 or 3 years with growers and we tell them, well, we think we're at a point where we can start reducing insecticide applications. Let's wait to apply the product until we see the pest. And we never see it, or we only see it very late in the season, and we might put on 1 or 2 applications instead of 5. So everything is context dependent, but we do not begin by making the recommendations that you stop all pesticide applications. That on the scale of farmers that we're working on, that would not be a wise recommendation. Question from John. Elon Musk has created an X Prize with $100 million in prizes for ideas that show direct carbon sequestration. Are you familiar with this? Yes, I am. I've heard your discussions of raising organic matter in soil and believe you may already have the answer. Please tell us you will work on this. John, I'm not alone in this field, and there are many, many people in the regenerative agriculture space who know that the answer lies with photosynthesis and grazing, regeneratively managed grazing, and so forth. And certainly I've been able to connect some dots to describe how photosynthesis can actually be much more efficient and much more effective than is commonly understood. But to answer your last question, I'm not actively working on it at the moment, and I'm not sure that regenerative agriculture answers are desirable by the team administering the X Prize, only because they're not a technological solution. I could be mistaken about that, but from what I've observed so far, there's a high probability that a technological solution that is not regenerative agriculture oriented will likely be the winner of the prize. And if there are indications to the contrary, I would be delighted to collaborate on a team to help bring about that reality. Question from Niels. Hi, Niels. We have a crop of Brussels sprouts that have an infection of gray aphids. Soils are light and the summer has been dry. So granular nitrogen that was applied in the summer is only now flushing, only now becoming available. SAP analysis were held up at customs, and so they're now perished and were not analyzed. The Brix in the upper leaves is 5. The Brix in the lower leaves is 6. What foliar materials would you suggest to improve the health status to the point where aphids are repelled? Niels, I'll give you an answer in the context of AEA products, and then you can look at labels and look at nutrient applications that could match that. I would suggest 6 quarts per acre of Photomag and 2 to 4 quarts per acre of Rejuvenate. That gives us a broad spectrum of magnesium, sulfur, molybdenum, boron, and also gives us a shot of sugar application to potentially overcome any stress that is limiting photosynthesis. That would be the approach that I would take and that I would have very high expectations for. Question from Don: how would a small grower use and apply BioCoat Gold? BioCoat Gold is an inoculant that can be used as a seed treatment. You can coat it on your seeds before planting. You can spray it onto the soil. There's many different ways you can apply it. Question from Gerald. Hi, Gerald. On various tree and vine crops, we've applied molybdenum either through our foliar blends or in our nitrogen plus humic fertigation blends. In our NCC SAP tests, moly starts out very low all season, then all of a sudden, some fields will jump from 0.3 parts per million up to 5 to 10 parts per million. When NCC reports anything above 1 part per million for example, is excessive. If this is is this high of a molybdenum level a concern? The answer is no, it is not. Is there anything we can do to offset it when it gets this high? And is there a management approach to help prevent high molybdenum like this? I'm a fan of higher levels of molybdenum like that. I believe, we have set different desired values on our SAP reports than what NCC's desired values are. And they vary from crop to crop. But if I recall correctly, I want to say that our desired values are usually in the 3 to 5 parts per million range. So we would not consider that to be excessive at all. And even at up to 10 levels, 10 parts per million, we would not consider that a problem. If molybdenum gets to be really high, and what defines really high varies from crop to crop, but I would say in most crops, based on we haven't actually experienced and observed this very often, but I would anticipate somewhere in the neighborhood of above 30 parts per million, between 25 and 30 parts per million and greater. The levels can become high enough that they can actually induce a nitrogen deficiency. I've only observed this a couple of times. It's relatively rare. Something else to think about is that in the case where you are applying molybdenum to the soil, molybdenum absorption is known to rapidly increase at higher pH levels. So there's a question in my mind, and I don't know the answer. Is there something that you did to alter soil pH, or maybe stopped irrigation water treatments or something that caused an increase in soil pH that caused a flush of molybdenum availability? That's one possibility. There's also other possibilities with bacterial population die-off, all of a sudden releasing molybdenum. There's various things that could be going on. Your last sentence: during rapid crop growth, it's like we can never catch up. Then once growth slows down, molybdenum jumps way up. The levels get high in both the old and the new leaves very equally. So that could actually, that could point to the last observation that I made, is if you have during the vegetative growth stage, when you have very rapid growth, there's a possibility, this is just a hypothesis, I could easily be mistaken about this, but my hypothesis would be that at that period, you're also transmitting a lot of sugars to the soil profile, and the bacterial population is expanding very quickly and consuming a lot of molybdenum. And then holding a lot of molybdenum. And then once vegetative growth slows, that bacterial population growth slows as well and releases a lot of the nutrients that they are holding. And that's perhaps why you are getting the flush of molybdenum. You can test that and play with it. I don't know for sure if that's the answer or not, but that might be something worth looking at. I know that we're up against the hour, but lots of people have asked questions, and there's lots of really good questions here. So I certainly understand if you do need to drop off, but in respect for everyone who's here, I'm going to continue and try to answer all the open questions that I can here. It's a question here that says, bicarbonate levels are high in our irrigation water. Therefore, we do nitric acid dosing. In this case, nitrate levels in our irrigation water is unintentionally high. This is a problem for soil health. Yes, that is a problem for soil health. And I would suggest that one of the very effective tools that you could use would be to add humic acids, add humic substances to bind that nitrogen and prevent it from being as rapidly absorbed. And there's an I think there's at least one or maybe a couple of webinars that I've done on this topic in the past that I would refer you to. But you can apply and inject humic substances into the irrigation water as well, so that they bind this nitrogen, complex it, and not have the plants absorb it to excess. Question from Damon. I'm in the process of completing my harvest. I practice no-till, chop and drop cover cropping. My soil is littered with plant residue. I live in a very sunny environment with low rainfall. When is the best time of day to apply the AEA fall soil primer? When you have time is the right answer. So we apply it at any time, morning, afternoon, evening. It doesn't really matter. I would say it's perhaps you might slightly increase the effectiveness of it if it's not applied or if it's not exposed to extensive sunshine right when you're applying it. But one of the tricks to the soil primer, the combination of biology, much of soil biology is susceptible to UV. So it's helpful to have a dark solution that the sun doesn't shine through the solution or shine through a droplet. And that's one of the reasons why we add Rejuvenate and Sea Shield, is because it gives us a dark solution. Question from Steve. You've got minerals listed on the plant health pyramid, but when, where, and how do you look to address calcium? This is a starting point for a lot of other agronomists. And it's a good observation, Steve. And I would say that it's also a starting point for us. It's kind of we don't make this or communicate this explicitly, but it's almost as if it's almost an assumption that this is such a foundational starting point that you have to start with calcium before you can really address any of this other stuff. Particularly when we look at getting nutrients and developing stable soil environments that deliver crop nutrition, so that we don't need to do it from a fertigation and foliar perspective. So I would agree that it is foundational, and it's where you really need to begin. Question from Davis. I'm trying to improve degraded soil with a low CEC, around 5. In listening to other lectures of yours, I've learned about increasing stable soil carbon. Should increasing the stable soil carbon be a main focus for improving soils with a low CEC? I don't think so. Because you have a low CEC, which translated means you have a relatively sandy soil. A relatively sandy soil is going to have a high gas exchange, which means it's going to oxidize very rapidly, and it's going to be very difficult to build up high levels of organic matter that is sustainable. You can certainly do that over time. If you look at a 20 or a 30-year, 40-year time horizon, you can increase organic matter in sandy soils up to several percentage points. But in the short term, what you need to produce amazing crops is not high organic matter. What you need is high levels of microbially active carbon. In other words, in the words of Christine Jones, liquid carbon or sugar. And so that is when you have these high levels of microbially active carbon or sugar in the soil profile, that is how you really sustain active biology, which is going to give you the energy to grow a really healthy crop. So that's the short-term answer. Building elevated levels of organic matter is going to take decades in that type of a soil environment and climate, and is not a worthwhile short-term goal. Question from Gurprit. Could spotted-wing Drosophila on blueberry be managed through nutrition? If yes. So yeah, the answer is yes. And it is exactly the same as managing. The short answer is you can't guess. I can't tell you that you need to address manganese and copper and zinc and magnesium and sulfur and expect you to be successful. That is not going to work. The only way it's going to work is if you actually use a SAP analysis to measure what you are low in or what you have excesses of and manage accordingly. So the answer is yes, it can be managed through nutrition, and it's the same nutritional profile of other larval insects or aphids, for example. So it's magnesium, sulfur, molybdenum, and boron. However, You will have to be consistent with it enough to the point to get good mineral balance in the fruit and in the berries, not in the leaves. So it may take more time, but it's entirely possible. Question from Michael. Hi, Michael. What is the minimum and maximum level of electrical conductivity, EC, in the soil you strive for when you measure EC with a direct soil meter? Do these values change during the different growth stages of a plant? Are these values different for different crops? And do these values change during the year with respect to different planting dates of the same vegetable during the year? Michael, you ask some great questions. I'm trying to think of how I could give you some specifics, but the specifics change in different soil environments. And so rather than giving you specific details, and I'd be happy to pick up that conversation with you, I would just say for right now, when there are two completely different models of how plants absorb nutrients. Are plants absorbing nutrients through the rhizophagy process and through bacterial populations or through simple ions? And this is important because when you have really healthy soils, as in really healthy microbial populations, abundant biology, you can have abundant plant nutrition with a much lower EC. So you can have electrical conductivity levels in the 200 to 400 micro CGS range and have abundant plant nutrition because the levels of soluble ions in that type of soil are low, but the levels of available energy to the plant are high. So there is this modifier. We can't think about EC just as being straight electrical conductivity of ions. We also have to consider what is the contribution of microbial metabolites and recognize that they can add a lot of energy to plant development that does not show up in electrical conductivity. So that's could be an entire discussion and webinar in and on its own, but I'm sure you can run with it from there. Question from Sarah. Have you used foliar nutrition much instead of chemical desiccation? If so, could you share some positive and negative experiences after tools to support producers moving away from glyphosate? The one tool that I really have experience with is using boron, specifically boron attrition, and not as a desiccant, but as a nutrient. And one of the pieces that boron does particularly well is it speeds up senescence and maturity, and it speeds up sugar transport from the leaves into the sugar sinks, which at the time of maturity, the primary sugar sink is the seeds. And so we are often, we've got a fair amount of experience with growers putting on foliar applications of boron, or actually, if you look at it systemically, you can also put on boron at the beginning of a growing season at planting or before planting or at any stage during plant development. And it will have this positive effect of speeding up senescence and speeding up maturity. And of course, the other piece that we have to consider is one of the reasons we have delayed maturity is because of over-application of nitrate nitrogen, or I could just say over-application of nitrogen that ends up being absorbed in the nitrate form. So the reality is that when you manage nitrogen and boron and some of these other nutrients well, you can move maturity forward. It's, again, crop and context dependent, but it's safe to say in many crops, you can move maturity forward by as much as 7 to 14 days. It can be really significant. Question from Franz. Farming macadamias in South Africa. I find your webinars extremely informative, like the bud optimization management. These tend to be focused on deciduous trees, however, and I tried to apply it on evergreen trees. The big difference being the lack of a rest period and consequent continuous photosynthesis. Will you have a webinar on the nutrition of evergreen trees sometime? So we could do that, Franz, but think of it as, if I'm not, we do, the only evergreen crop that we work with that I'm familiar with or can think of off the top of my head would be citrus, I guess avocados as well. So think of it in terms of the reproductive cycle. Because even while they are evergreen, there is still a distinct reproductive cycle. When does bud initiation happen? When does, or when is the peak? Maybe that would be a better way of saying it. When's the peak of bud initiation? When is the peak of blooming and pollination? When is the peak of fruit ripening and maturity and so forth? And manage according to those peaks. And our webinar and our course on critical points of influence will address all of those, and you can transfer that very directly. In a question from Maj, in a healthy plant, some nutrients should be lower in the older leaves than in the younger leaves. Think about that. Some nutrients should be lower in the older leaves than the younger leaves. I'm trying to think of specific examples. The sap analysis of that healthy plant reflects such a result. How does NovaCrop Lab interpret the results, say, on the old leaves should have a higher nutrient concentration than the young leaves? This is a contradiction observation. Please explain this discrepancy. I'm not quite following your question, and there must be something that I'm missing here. And so our experience has been that there are some nutrients which will accumulate in the older leaves. Particularly, the two that come to mind are calcium and magnesium. Because of photorespiration, those two nutrients tend to accumulate in the old leaves, and as the season goes on, the levels in the old leaves can be as much as 2 to 3 times higher than in the new leaves. I don't think, to your first sentence where you say in a healthy plants, some nutrients should be lower in the older leaves than in the younger leaves. Unless there's something I'm missing, I can't think of any instances where I would agree that that statement is correct. It happens occasionally, particularly in the case of phosphorus. It's often the case that phosphorus moves from the old leaves into the young leaves and is therefore much higher in the young leaves, but not that it's particularly healthy for that to be the case. So possibly I'm misunderstanding the question or just missing something, and if so, please follow up. But those are my thoughts at the moment. Question from Nigel. Hi, Nigel. It is suggested that amendments made by fermenting plants or extractions of bones and shells using vinegar produce amendments that have minerals in forms that plants can use directly. And I would say, I would agree with that. And in general, I would say the answer to that is yes, because if you're doing using vinegar, natural vinegar, it has an abundance of free electrons and is very reduced. And so it would have both a reducing effect and a chelation effect. The analysis of these products reveals broad spectrum content of macro and micro minerals. Would you please comment on the ability of plants to utilize these amendments directly? Are they in the ionic form that plants can use directly? And my understanding is yes, they are, and that I would expect plants to absorb them very quickly and use them very quickly. Question from Jim. I'm in northern Wisconsin farming sandy loam soil. I've heard you discuss keeping cover crops vegetative to maximize carbon and sugar production and transfer into the roots and soil. And I also have a problem with wild radish weeds. I like to flail mow the cover crops after the weed seed has set to keep the plants vegetative and prevent weed seed drop. But driving on the fields increases my compaction. Can you discuss what you consider when you're trying to decide to flail mow, even if it increases compaction? Oh boy, Jim, that's a very specific question. And for for the benefit of everyone else here, and hopefully also for you, I would ask two questions. One, is it possible that you could graze instead of mow? And if the answer to that is no, the second question that I would ask is it possible that you could get a lighter mower? That would be where I would start. And if neither of those are practical, then we may need to figure out how to deal with the compaction in other ways, considering you have sandy soil. What about, actually, there's something else to consider as well, which is why are the wild radishes such a significant problem? And perhaps one solution to that, why do they desire to grow there? One possible solution for that might be to consider using oilseed radishes as a cover crop, both to help relieve the compaction and to address the soil issues that the radishes are there to help solve. So that could have a double beneficial impact at the same time. Question from John. Our sap analysis on our tree fruit have had increasing levels of total nitrogen above the recommended levels over the last few years. We have maintained relatively low nitrate and ammonium levels during this time. Is there a limit to how much complete nitrogen forms we have in the plant? It's a fascinating question, John. I'm not aware that there is one. As long as the nitrogen is in complete forms, it's in the form of enzymes, or it's basically, if it's not in the nitrate or ammonium form, and if it's not increasing disease and insect susceptibility, then from a practical perspective, I'm not aware that you can have excess of proteins in a plant that have negative health consequences. Not saying that there aren't any, but I don't know of any right now. Question about biology. Are there any manganese reducing bacterial strains ever noticed in soil? The answer is yes. There are many strains of bacteria that reduce manganese when soils are anaerobic. Or in other words, when soils are saturated, when soils are wet. Even many common strains of biology that can also be oxidizers. In other words, those that are the facultative aerobes or facultative anaerobes have the capacity to reduce manganese when the soil is saturated. So it's a common capability. Ian Thomas followed through with the questions you asked at the beginning. Ian, thanks for being here with us. Yes, I answered because your questions came through on email. They were the very first ones that I answered right at the opening. So I'll let you catch the replay on that. Question from Elmer. If there is no lab available to make sap analysis, how can we on farm get a better idea of what is going on? Ship to somewhere that there is a lab available, I think is the, this is interesting. I never signed up to be the champion of sap analysis. So it's interesting that I find myself just having this conversation over and over again. Or I should say I never deliberately signed up. I've kind of become the champion of sap analysis by default, it seems. There is no lab available in the US or in Canada. I'm not sure where you're farming. Well, let me qualify that. There are labs that are seeking to develop their own version of sap analysis with varying degrees of success here in North America and in different places around the world. And I care about getting results, and I care about having reliable information. When we're working with farms who have a great deal of crop value at stake, I'm not comfortable guessing about making recommendations. And so as a consultant, as an agronomist, I am not comfortable making recommendations without sap analysis. And we can make lots of recommendations based from experience. But every time I think about making recommendations from experience, I realize that those experiences, what we have experienced, has been so valuable and so powerful because we have had SAP analysis. And so for me to make the recommendation for you that you can do it without SAP analysis is it's difficult for me to make that recommendation. Is it possible to do it with SAP analysis? Without question, yes, absolutely. It's going to take a lot longer and you're going to make a lot more mistakes and spend a lot more money than if you did have SAP analysis available. So I know that there are growers around the world, I have no idea from how many different countries, but quite a number, that export samples to Nova Crop Control in the Netherlands for analysis because of the value of the information that it brings. How can you get a better idea of what is going on? You need to become really good at visual analysis. You need to learn what different plants are telling you. Get the books Weeds Control Without Poisons and Weeds When Weeds Talk by Jay McCammon. You need to learn to read the landscape. And of course, learning to read the landscape comes from a lot of education and a lot of experience. And the value of SAP analysis is it speeds up that learning and that education process by at least an order of magnitude. So, yeah, that would be the way that I would answer the question. You can also use dry matter-based tissue analysis, but just understand that it has lots of limitations that the SAP analysis doesn't. Question from Stephen. You've mentioned in a previous webinar on preventing nitrogen and phosphorus leaching that you worked with a group of small-scale fruit and vegetable growers who were using large quantities of sugars with their nitrogen and achieving outstanding results. Is there anywhere I could learn more about this, or could you expand a little on application rates and the kind of results they achieved? I don't recall exactly what I was referencing there, but this is a relatively common application for us in our consulting work. The applications you asked about, could I expand a bit on our application rates? We are typically running between 3 and 5% of the nitrogen solution as rejuvenate in these situations. So not 3 to 5% of the N number, but 3 to 5 pounds or 3 to 5% of the nitrogen solution. And this is an important distinction. So let's say you have a liquid nitrogen that is, for the sake of discussion, we'll use liquid 32. So we do not use 5% of 32%. We use 5% of the total volume of the liquid 32. And the reason for that is because nitrogen is present in this solution as either nitrate or ammonium, or in some cases urea, which would be ideal. And so when we calculate adding humic substances or adding sugars, it is in proportion to the nitrogen compound, not the actual N atom, because we are complexing or chelating or metabolizing and converting nitrate and ammonium or urea, not the N atom itself. Hope that makes sense. Question from Travis. Have you had any experience managing Cercospora leaf spot in sugar beets to keep this fungus suppressed enough to not cause economic damage without the excessive use of fungicide? I personally have not, but I know that a couple of our agronomists on our team have had. Question from Joe. I have a plasma tech company that produces pure nitrate and nitrite in the solvent. We are presently adding a micronutrient. So Joe's question is a combination of self-promotion and a questioning about whether foliar applications of nutrients can regenerate or can suffice to regenerate tree health in citrus while improving soil fertility as a foundation. And the answer is yes, that's the foundation of our approach at AEA. Steve asked the question, can you talk about amino acids, the Krebs cycle, and why we use citric acid or the benefits of it for chelation? Steve, that's like asking, can you talk about the known universe and cosmology? It's kind of everything tied together there. Um, I'm going to hold on to that question and come back to it at the end because there's too many possible connections there and too many different directions we could go in. Question from Levi. Brand new to this entire world of farming and orcharding. I would like to begin to learn more about disease and plant health management, specifically with apples, peaches, blueberries, grapes, and blackberries. Do you have books or other resources you recommend to start learning about this? The answer is yes, I do. And I used to get asked that question so often that I actually wrote out a list and posted it on my blog. So if you just do a search for John Kempf recommended reading list, it should come right up for you. And there's a list of, I'm not sure, probably 40 or 50 books that I recommend very highly. Question from Darren. In your experience, have you seen Eutypa or bacterial canker be reduced or prevented in apricots or cherries? And the answer is yes. In fact, I've seen both of those be completely reversed to the point where we had some orchards that were severely infected to the point where the grower was prepared to bulldoze them out and replace them. And we completely reversed it to the point where they became some of the top yielding blocks in the orchard. And that I should clarify that was specifically on cherries. Have less experience on apricots, but I'm sure the same is achievable. And if you were to ask me, what did we do to achieve that result? The answer is, I don't know. We did everything. We managed nutrition and biology differently. In broad brushstrokes, the common practice was to apply high levels of zinc, and we found, amongst other things, and we found that zinc levels were excessive and manganese and copper and boron levels were extremely deficient. We changed that around. And just the combination of everything that we did, bacterial canker reversed itself from being severely infected to non-detectable infection that was validated by a university and by a field day of 100 plus growers who were present and were unsuccessful in finding any. And we still have 20 questions left, and we still have about 80 people here. I'm still going to continue going through these questions. However, I do need to take a very quick bio break. So I am going to take 20 seconds, maybe 30 seconds, and I'll be right back. My apologies. Hi, friends, I'm back. I think I stayed pretty close to my 30-second mark. Question from Tom. Can you give us a run through of your nutrition wheel and the difference of order of importance of the nutrients in the wheel? If there's not enough time today, do you think you could do that in a full Zoom? All right, so Tom, I've actually put together an in-depth course on this. It's on the Academy and it's on Kind Harvest. And it is titled the Precision Ag Plant Nutrition Management course. So there is a course where I spend, I'm not sure, 3 or 4 hours or so going through the nutrient interactions and describing how we manage them and how we make recommendations from them. And if you've already seen that and you have any further follow-up questions, then I'd be happy to follow up with you and see if we can dig into some of those more deeply. Question from Stephen. In cannabis grows, they use what they call living soils, which are very complex organic substrates. Do you think this approach could be effective for growing container strawberries, raspberries, and blueberries? What pitfalls can I expect to run into? So the answer is yes, it can be very effective, and it is already being used. We've been using this approach for some time with blueberries and raspberries. And I know it's being used in the Netherlands quite a bit with strawberries, if I'm not mistaken. So what pitfalls? The only one that comes to mind off the top of my head is that you need to manage water differently, because many of these substrates do have a much higher water holding capacity than something that is peat moss and perlite based. Question from Timothy. For high value fruit and vegetable crops, do you recommend using the fall soil primer more often? Before every seeding and or transplanting, do you recommend more frequent applications of Spectrum and or BioCoat Gold through the irrigation system in these systems? The answer, Timothy, is it depends. Sometimes, yes. We actually have There's different regions. We have soil challenges where growers prefer to apply the soil primer, the combination of rejuvenate and spectrum, consistently through the growing season. In some cases, they're putting on microdoses, smaller doses every 4 weeks, or in a few cases, even as frequently as every 2 weeks. So there is more and more of that happening with higher value crops who are spoon feeding that throughout the growing season. And so there is value in that. But the question is, what's the context? In cases, let me say it this way, where you have really challenged soils, the more challenged the soils are and the greater difficulties that biology has to overcome, the greater the value in those spoon feeding applications. The goal, of course, is to eventually get to a point where we need fewer applications or no applications at some point in the future, depending on the rest of the management system. So, question here on what kind of protocol should be applied to control bacterial canker in sweet cherry. And I already addressed this a little bit. And my honest answer is I can't point to 1 or 2 nutrients or 5 nutrients and say we did this and bacterial canker went away. We used sap analysis and we balanced plant nutrition and bacterial canker went away. It was the sum totality, as far as I know, it was the sum totality of everything that we did, not any one thing that we did. Question from Mike. I collected compost from a Johnson Sioux bioreactor recently. Lab analysis reveals a nitrate level of 1,800 parts per million from the sample. Sample is composed of oak, hickory, and maple leaves and lawn clippings. Any suggestion for interpreting this result when using compost in garden and with perennials? Well, first of all, I would say that 1,800 parts per million in a compost sample is comparatively low. That would be high if it would be considered to be high if you were measuring soil, but we're not measuring soil, we're measuring compost. And so that level is comparatively low. And I would anticipate, based on the sources of raw material that you identified, that you would have a very wide carbon to nitrogen ratio. And so I would not consider that compost to be to be a source of nitrogen for a growing crop. Very minimal at best. Question from Steve. What is happening when we have higher Brix in the old leaves than we do in the new leaves? What is happening is that the plant is not transporting sugar well, and that could be for a couple of reasons. Or it's the plants are not metabolizing sugar in the leaves very well. There's 3 possible reasons that come to mind. One is that the plant doesn't have adequate levels of boron, and it's not moving sugars out of the older leaves up into the upper parts of the plant very well. That's one possibility. The second possibility is that the plant has low levels of potassium, and the same thing is happening. The plant is not transporting sugars very well. The third possibility is that the plants have a deficiency of phosphorus, and phosphorus levels, or did have a deficiency of phosphorus in the past, and phosphorus levels are low on the old leaves, but high on the new leaves. And because of the phosphorus deficiency on the old leaves, the plants are not metabolizing sugar very well. I've observed all 3 of those to occur at different times. So it depends on what levels and ratios you're talking about. But from a nutrient interactions perspective, those would be my first guesses. And it's also worth thinking about water. Do the plants have water stress? Are they drought stress or something like that? That might also have an influence on sugar transport and metabolism capabilities. Question from Stephen. I winter lettuce in tunnels. I'm finding it difficult to raise the Brix above 3. The plant is quite balanced in the sap analysis, but I'm in a cloudy area. Is this simply a lack of sunlight primarily? And the answer is probably yes. Could also be slight contributions of carbon dioxide, but I would guess it's probably sunlight is the first place to look at. And depending on the type of production. If you're in a heated greenhouse in particular, it might be worthwhile looking at putting in some full spectrum LEDs or putting in lighting that of the correct wavelengths to increase photosynthesis. Question on how to increase the soil health and the quality of bell peppers. Current problems are that there are wrinkled and dehydrated skin, and sometimes having corn borers inside the bell pepper. Highly saline soils, calcareous clay, loam soil with compaction issues, soil cracks after irrigation, plow pan, 15 to 16-inch depth, underground water table, pH of 8, high levels of potassium, relatively low levels of phosphorus, and organic matter of 0.5%. So the symptoms that you describe on the fruit themselves, which is what in the situation you're describing, we have to begin by looking at, by considering the plant as the final report card. What's the plant telling us that is really going on? And the plant is telling us that it has low levels of calcium and possibly excessive levels of potassium. So you've mentioned that you have calcareous soils that are highly saline. So when you have that high level of salinity, the plant is probably absorbing sodium and it's absorbing potassium very readily because those are both quickly absorbable. It's probably also absorbing bicarbonates and chloride, and as a result of accumulating all of those other cations, it's not accumulating calcium very well. So I would suggest you don't indicate whether, actually you do say that soil cracks after irrigation. So I'm presuming you have some irrigation. And in that context, I would suggest first of all, you need to reduce soil pH to increase calcium availability. And so in this type of a challenging environment, it's not so much about investing a lot of money in changing the bulk soil pH, but you need to change the pH right in the root zone. And to do that, if you are using drip irrigation or whatever form of irrigation you're using, I would suggest adding acidifying agents to the irrigation water to help develop a root zone that allows for calcium absorption and better absorption of other nutrients. That would be the place where I would begin, because I suspect that the majority of the problems that you identify are a lack of available calcium. Even though you have calcareous soil, you have abundant levels of total calcium in your soil profile, there's a high probability that the available calcium is actually very low. Question from Noel, sphagnum peat moss. We have tons of it in many large garden beds. We have heard you mention how biology, especially fungus, does not work well with it. Are we better off starting over and replacing all the soil in our beds? We also have perlite mixed in. We created and filled these beds before we discovered regenerative practices. What kind of issues do you see us running to in the future if we decide to leave it in and try to live with it? It is possible. So first, you ask a couple of different questions. One is, it's possible to add amendments to allow mycorrhizal fungi and other fungus to thrive in a peat moss substrate. Humic acids are particularly good at that. I would suggest adding some dry humates or some different humic substances like humicarb. And you can try to balance that out. The one downside that you should be prepared for is that as peat moss ages and decomposes, it releases very high levels of potassium to the point where you can readily produce potassium. I shouldn't say potassium toxicity, but potassium becomes so high that it creates deficiencies of calcium and other elements for many different crops. So you should be prepared for that. As the peat moss ages and decomposes and breaks down, you should particularly try to focus on growing crops that have high potassium requirements to pull some of it out of that soil profile. It'd be a great substrate for growing potatoes, for example. You could mine tremendous amounts of potassium based on your crop selections and take it out. So that would be something that you should keep in mind. Questions about applying sugar. What sort of sugar are you considering? While different sugar sources have slightly different impacts on plant metabolism, the foundational issue that we're trying to address is that they simply don't have enough energy, period. And so we typically, we commonly use our preferred form of foliar application is rejuvenate, because it contains not just sugar, but other enzymes and other materials as well. But it can be any form of sugar, I think. There's some slight differences in effect that have been documented, but I'm not aware of any differences that are significant enough to make it worth a lot of extra effort. Question, how do you detoxify boron in high boron irrigation water? Rather than detoxifying it, I would simply say that you counter it by providing abundant levels of available calcium. When you have high levels of available calcium for the plant to absorb, you will not see any problems with boron toxicity. Question from Kent. Do you find that providing soluble nutrients to plants keeps them from feeding soil microbes or disturbs the natural nutrient cycle? The brief answer is yes. In the case of nitrogen and phosphorus, that effect is very well documented in the peer-reviewed literature, and actually in the case of potassium as well. So the short answer is that if you provide soluble nutrients, then the biology doesn't have to work and the plant doesn't have to work, and you break that relationship between plant and soil biology. Question from Elmer. What can we do after a muddy and late root crop harvest that is past the winter wheat seeding time to mitigate the negative effects of the harvest process? There's a lot of context missing there for me to be able to give, to have a qualified opinion. But I would suggest as one possibility to consider is to consider seeding rye instead of wheat, because rye will germinate. I mean, rye will even germinate after there is snow on the ground and after it's been frozen. We have here in our region of Northeast Ohio, there have been many times where we harvested really late and we planted rye and the soil froze a couple of inches deep and we started getting snow, and we would get anywhere from 10, 12, 14 inches of snow accumulation over a period of a month or two. And after a month, by beginning or mid-December, the snow would melt and we would have a bare period, and the rye would have germinated and be growing under the snow up to 1.5 inches to 2 inches in height. So rye will germinate and grow in very challenging conditions that winter wheat will not grow in. So that might be something to consider. Follow up from the earlier question on peppers. To continue, we flood irrigate. Should I spray elemental sulfur in autumn, winter to reduce overall soil pH, or is there any way to reduce soil pH? So yeah, you could consider spraying elemental sulfur or just broadcasting elemental sulfur as well. And again, context dependent of what are your levels of alkalinity and how much free limestone do you have in your soil profile, but just bulk applying elemental sulfur is probably going to be your least expensive application if you have that available. So I think that wraps up all the questions that we have so far. I see a number of you have stayed here for the entire 2 hours. I want to say thank you. I hope you found it useful and valuable, and I look forward to speaking with you again soon. Thank you for the really great questions, by the way. The questions were really great. There were a few that I didn't answer because they were kind of repeats or overlap with what I had asked earlier. But it's been a wonderful conversation. I've enjoyed it. I've enjoyed the interaction and look forward to speaking with you more again in the future. Have an awesome day. Happy growing. Thanks, everyone. Be well.